Hello, this is Bahandra Didia. In this portion of the book on liquid formulation, we continue to focus on solution dosage form. This is the eighth module and builds on information from pre-formulation, composition and formulation, equipment and processes. And in this module, we have two topics, quality of the product and clinical study formulation. It is common practice to have different level of formulations based on the study phase. Quality of the product must be assured that product meets the standards. Quality is built into the formulation. Readers referred to the submodule video 2.3 physical chemical on quality, stability, and chemical kinetics. In this submodule related video on quality and stability protocol, we develop understanding of quality for solution dosage form. Quality of the product is established by three related criteria. Quality by design, refer to module 2.3, physical-chemical characterization, quality, stability, and chemical kinetics. Quality control, testing of ingredient and finished product and in-process testing done in laboratory. Stability studies of the product are designed following the regulations and industrial standards. We'll look at quality by design, quality control, and stability studies for solution dosage. Quality should be designed into the product. Problems related product design. That is, we don't build product with quality by design. QBD is a systematic approach to development. It has a predefined objective. It emphasizes formulation. Quality control testing is based on the science and product quality risk mitigation. When we develop specification, it must address the risk. QBD together with finished product testing and ongoing stability ensures product quality of each lot. Let's look at QBD. Quality by design framework starts with QTPP, product profile, kind of dosage. Critical quality attributes for each product are established based on the process. We design the space conditions. For example, mixing time, temperature, material characteristics, particle size, design to address CQA. Based on that, we develop control strategy. Control strategy could be tighter manufacturing condition, quality control specification, or could be tighter material control, particle size specification. We verify that and validate. So let's look at quality by design for liquid and solution product. We talked about critical quality attributes, CQA, critical process parameter, what are the process conditions, and CMA, critical material attribute. We evaluate CMA and CPP to characterize the critical quality attribute. We establish initial quality risk at the beginning of the developmental product. And then as we develop a product, we mitigate what are medium and high risk into low risks. That can be done many different ways, including quality control testing, resolving critical material attributes, or critical process parameters. CMA, polymorphic form, are critical for dissolution, particle size, product to dissolve faster. Surfactant help with the product if product is hydrophobic. And its concentration will also provide information on micellar solubilization. Preservative types and concentration important for solution dosage form. Unionized form of preservative is generally more effective. Solvents, co-solvents, buffering agents could be critical materials, and their properties will affect the solution dosage form. Let's look at some of the processes. If we say parenteral product Then sterilization in place. How are we sterilizing this bulk material? pH of the product range, headspace or oxygen sensitive product, provide nitrogen headspace. Mixing speed and time, extremely critical. Engineering aspect, impeller types and design. We talked about this in earlier module. If this is not properly situated, it will not allow for mixing. Heating and cooling and temperature that can be controlled much faster. Homogenization and filtration steps, again, very important. CQA, critical quality at pH product, and how is that adjusted? The solution, solubilization portion of it, how fast it dissolves. For example, particle size would be affecting that. Viscosity, that would depend on viscosity agents are. Microbial limit, obviously preservative type and concentration will be important. If the product is sterile, then sterility and endotoxin levels become very, very. So we assign low, medium, and high CQA and then develop a control strategy. Quality by design attributes: physical, chemical, microbiological, biological, and soft products. General physical attributes would be clarity and crystallization. Will the product precipitate? Viscosity, density of the product, color, and cosmetic and pharmaceutical. Very critical for oral dosage form is palatability. Many of the drugs in some dosage form are bitter in taste and may not be acceptable. So palatability, flavor, and taste become very critical. Pourability for oral solution, dosing device, dose uniformity. In earlier module, we looked if we adjust density of the product, how the dosing device uniformity is complied with. Weight control during manufacture, packaged product, weight and fill volume, extractable, leachable from the container closure systems. All the quality attributes, depending on product, they could become critical quality attributes. Chemical attributes are active, identity and assay, impurities. What are the impurities? Assays of the preservative, alcohol, and similar volatile compounds. We need to assay them to make sure that they are at the concentration required in the product. Microbial attributes, solution product needs to be preserved. Microbial attributes needs to conform to USP or regulatory limits. Biological assay, for biological product, there are other assays we need to make sure they are acceptable. Sterile products, sterility, pyrogenicity, and osmolality, and many of these could be critical quality attribute, again, depending on the product and formulation. During the development, minimize critical quality attributes. Let's look at QBD, CQA risk for initial and final risk as reported by FDA during approval of the drug. The drug substance is light yellow and greenish yellow. Powder is used and pharmacists reconstitute powder with purified water to provide an oral solution at 0.75 milligram per ml. This is the product looks like as a label. It's given to the consumer as an oral solution, 60 milligram per 80 ml, 0.75 milligram per ml. As drug active, mannitol is a sweetener, also gives you soothing oral feel. Isomalt is a sweetener, tartaric acid pH, sodium benzoate preservative, PEG 6000 for density and viscosity. So the solution is pourable or it's withdrawable by the syringe. Sucralose, another sweetener, ascorbic acid antioxidant, disodium EDTA as a chelating agent and flavoring agent, a strawberry flavoring. In earlier module, we looked at these type of composition containing sweeteners, pH, preservative, antioxidant, chelating agent, and flavor and density and viscosity adjustment. The reason for reconstitution with purified water by a pharmacist to prepare oral solution is product active is unstable. Shelf life of a reconstituted oral solution under refrigeration conditions only 64 days. At room temperature, it's only 5 days. And pharmacists will explain that to the consumer. So let's look at the initial risk assessment. Formulation, container closure, process parameters, they were low risk initially. This is a reconstituted product. That means once you add water, that has uniform distribution oral solution. This initially was classified as medium risk. So this was reduced to adequate risk by requiring pharmacists to reconstitute with purified water rather than consumer taking it home and reconstituting it. Physical stability, solid state, formulation, raw materials, polymorphs, particle size, process parameters, they were looked at it, and initially they were classified as low. Palatability, drug has a taste, not palatable, and this was classified as a medium risk. Upon studies, it was classified as adequate additional sweeteners and soothing oral feel like mannitol. It was palatable. Microbial limit were looked at it from formulation, raw material, process parameter, scale up, equipment used at the manufacturing site. All these parameters were examined. Initial risk was low and final risk also was low based on the CQA risk, quality control, finished product specifications, and the product is approved by FDA. Initial risk is during development. As we develop the product, we look at the risk and start mitigating it. Quality control testing. The set of procedures to ensure that the particular lot of the product conforms to the established specification. Quality testing at each level of manufacturing, from development to commercial scale, is done on representative samples. Specifications are based on CQA, critical quality attributes during research and development, pivotal stability lot, and validation of commercial product. So in this video, you will see several instruments used in common testing in a pharmaceutical laboratory. This is a dissolution, HPLC, very common, or UPLC, viscosity measurement, titration, friability, disintegration, shaking, solubilization, dissolution sampling, and dilution of it, and supervisor. Expiration date and stability testing, or shelf life. For a oral solution product after reconstitution, shelf life was small. FDA has requirement for stability testing, the shelf life or expiration dating for drug product. Stability studies are similar for cosmetic. So for pharmaceutical, accelerated studies on finished products are conducted at 40 degrees. General duration is 1, 2, 3, and 6 months. Intermediate storage conditions, 30 degrees, 65% humidities, are done in case 40 degrees centigrade studies do not show adequate stability. Storage condition studies are generally done at 25 degrees and 60% relative humidity. Most programs are 0, 3, 6, 9, 12, 18, 24 months, and yearly after that. For solution dosage form, freeze-thaw cycle is done. Product is frozen at minus 10 degrees centigrade, exposed to ambient condition, and this is repeated for at least 3 cycles. Preservative effectiveness needs to be monitored throughout the shelf life. General protocol for stability studies at room temperature storage, 25 degrees C, 60% RH, and 40 degrees, 75% as an accelerated condition. Initially 0 time points, all the testing is done at, and this shows a representative testing. There may be dosage specific studies that needs to be done. They have to be incorporated in it. Products are kept inverted and upright. Reason for doing inverted is solution is in contact with the closure system.